All right, so again, this is part 2 of the chapter 13 recordings. Again, for meiosis, we use 4 phases: prophase, metaphase, anaphase, telophase, and of course cytokinesis. But instead of just leaving it like they're worded in mitosis, we add a Roman numeral. So that tells you which division, meiosis 1 or meiosis 2. So we'll look at prophase 1 because a lot of things happen in prophase 1 that you need to know. During prophase 1, all of the same things happen as what happens in prophase of mitosis. So you have chromatin condensing into chromosomes. You have centrioles moving to opposite ends of the cell. The nuclear envelope disappears. So all of those same things happen, but one more thing happens, and it's called crossing over. Crossing over is going to be exchange of genetic material between the homologous pairs of chromosomes. And it's going to increase the genetic variety in the offspring. In metaphase 1, instead of like mitosis, we would say that the chromosomes line up on the metaphase plate or at the middle of the cell. But in metaphase 1, instead of single chromosomes, it is pairs of homologous chromosomes. So I just say homologous pairs line up at the metaphase plate. And during metaphase 1, you have another event, which is not listed here, but you have something else that contributes to genetic variety, and it's called independent assortment. So we're going to learn about the how meiosis increases genetic variety. One of the things happens in prophase, it's called crossing over, and the other thing happens in metaphase, and it's called independent assortment. Then we have anaphase 1, and here it's worded differently than it is in anaphase of mitosis. In anaphase 1, instead of saying sister chromatids separate, we're going to say pairs, homologous pairs separate. Sister chromatids actually remain together, and they don't separate until meiosis 2. And then you have telophase 1 and cytokinesis, which are identical to what we've seen before. And this is what meiosis 1 is going to look like. You have the homologous pairs finding each other. Oh, I didn't even say that. I didn't see it in the notes. It says in early prophase 1, each chromosome pairs with its homologue, and that is called it's called synapsis, even though they don't have the that word on here. But each chromosome pairs up with its other member of the homologous pair. And that crossing over couldn't happen if they didn't pair up, because when they pair up, they're close together, and they can exchange or cross over pieces of their chromosome. Okay, that's what crossing over means. So you can see that here. You can see that homologous pairs have found each other. They've matched up, and they're so close together that the two sister chromatids, one from the mom and one from the dad, that are right beside each other, they can actually cross over and then exchange pieces of DNA. And you can see that in, I don't see it in the picture of metaphase, but if you look at anaphase, do you see how the little ends of the red chromosome are blue? Little ends of the blue chromosome are red. That's what happens with crossing over, is you get pieces of the other homologous chromosome, in its pair, in its twin or whatever. So still, you have metaphase is when the homologous pairs line up at the middle of the cell. In anaphase, the homologous pairs separate, and then telophase would be the same. Now we're going to have meiosis 2, which is really easy because it's just like mitosis. And in meiosis 2, you have prophase 2, metaphase 2, which in this case, in metaphase 2, the sister chromatids line up at the metaphase plate. In anaphase 2, the sister chromatids separate. So this is just like mitosis. And then telophase 2 and cytokinesis are the same as what we've learned. Those are the two that aren't any different between mitosis and meiosis. Cytokinesis and telophase are both the same, and it doesn't matter if it's meiosis or mitosis. All right, and again, at the end of meiosis, there's 4 daughter cells produced from one parent cell, and each daughter cell has only half of the chromosomes of the parent. So you can see that meiosis 2 looks like mitosis. The chromosomes, individual chromosomes, line up at the middle of the cell. Then the sister chromatids separate, and then you have telophase and cytokinesis. Now there is a Bioflix animation on meiosis in its slide 47, and you can watch sperm formation in slide 48. And then there's the word synapsis. Okay, so we're going to do the notes for each of the phases, but I'm not going to really, I just went over it, so I'm just going to highlight the things you need to know. So, prophase 1, you need to make sure that you know that a process called synapsis occurs in prophase 1, and that is when the homologous pairs match up. They find each other, and they match up. Now, for some reason, we're comparing mitosis and meiosis here. Right in the middle of it, just seems inappropriate to be doing that right now. But mitosis conserves the chromosome number because the 2 daughter cells are genetically identical to the parent. Meiosis reduces the number of chromosomes. And so that produces 4 daughter cells that are genetically different from each other and the parent. All right. Meiosis has these events that happen that are unique to meiosis that do not occur in mitosis, and all of them occur during meiosis 1. In prophase 1, we have synapsis, which is the homologous pairs find each other and pair up. And then we have crossing over that occurs at the same time, crossing over of pieces of the chromosomes from the homologous pairs. So both of those things happen in prophase 1, synapsis and crossing over. Then we have homologous pairs lining up at the metaphase plate during metaphase. And then we have separation of homologous pairs during anaphase 1. So all these things on this slide, which is slide 52, all of these events only happen in meiosis, and specifically only in meiosis 1, and they don't happen in mitosis. So you can compare for meiosis 1, you can see metaphase over here on the right is homologous pairs lined up in the middle, and then metaphase over here on the left of mitosis. Metaphase 1 of meiosis is when you have homologous pairs lined up. In metaphase of mitosis, you have all of the chromosomes lined up individually at the metaphase plate. And then going back up to prophase, of course, you have homologous pairs finding each other. That's called synapsis, and then crossing over. Now, when the chromosomes are separated during anaphase, in mitosis, it is sister chromatids that are separated. But in meiosis, it's homologous pairs that are separated during anaphase 1. So genetic diversity, the source of genetic diversity are mutations in the DNA code, changes in the DNA code. Mutations create different versions of genes, and a version of a gene is called an allele. An allele. So mutations create different alleles, different versions of the genes that are going to lead to different traits. So the original source of diversity is just mutations in the DNA that happen when DNA replicates. And the other source of diversity is crossing over during prophase 1, independent assortment of chromosomes during metaphase 1, and random fertilization, which means since all of those 4 sperm are different from each other and all of those 4 eggs are different from each other, it's random which sperm fertilizes which egg. So, that is going to generate some diversity too. This, now we're going to talk about independent assortment. And independent assortment means that we're just going to say that the blue chromosomes came from the dad of this parent cell, and then the red chromosomes came from the mom, okay? So if the blue chromosomes came from the dad, the red chromosomes came from the mom, you can see that they lined up where both of the dad's chromosomes are on the left and the mom's are on the right. This lining up or assortment of chromosomes is independent of the other homologous pairs. So if the top homologous pair lines up this way with blue on the left, red on the right, the bottom pair can also line up the same way, or it can line up with red on the left, blue on the right. And you can see how there would be so many more possibilities of combinations, of how they're lining up when you've got 46 chromosomes or 23 pairs versus only 2 pairs, So independent assortment is just the lining up of homologous pairs during metaphase, and how they line up. And then of course, the other source of genetic diversity is crossing over. So there is an animation on slide 62. It's called Genetic Variation from an Independent Assortment of Chromosomes. And I would highly encourage you to watch this one for sure. And then we have the random fertilization. So every zygote has a unique genetic identity because of mutations in the DNA, because of crossing over in prophase 1, independent assortment in metaphase 1, and the random fertilization. It's random which sperm fertilizes which egg. So all of that results in a huge amount of genetic diversity. Oh, these are cute. This is a rotifer, which is actually, it's a microorganism. I mean, you have to use the microscope to see it, but it's multicellular. It's technically an animal. These are really cool. I like rotifers. Yeah, we're at the end. Okay, so that's it. We've covered mitosis. We've covered meiosis.